Hello, hello friend. Welcome to our channel Quality Guru. Friend, our topic is revised schedule and requirement for sterile and non-sterile preparation. Schedule M was introduced in the year of 1988 as a part of Drug and Cosmetic Act 1945. On 28 December 2023, the Ministry of Health and Family Welfare released updated regulation under Schedule M of the DNC Rule 1945. Revised Schedule M includes the incorporation of pharmaceutical quality system QRM, that is quality risk management, product quality review, qualification and validation, and implementation of computerized storage system. As per the notice dated 28th of December 2023, the Ministry of Health and Family Welfare has released updated regulation under Schedule M of the DNC Rule 1945, making a major step toward improving adherence to quality standard. These regulations, which include the good manufacturing practices for pharmaceuticals, are intended to change the way that pharmaceuticals are made in India. Key changes in the revised Schedule M. Schedule M, incorporated as a part of DNC Act in 1988, helped to set foundation standard for manufacturing quality of pharmaceutical product. The revised Schedule M include 5 more categories to ensure compliance with global standard as follows: pharmaceutical quality system, quality risk management, product quality review, qualification and validation, implementation of a computerized storage system. Revised Schedule M in India mandates separate and comprehensive requirement for sterile product in Part 2 of the schedule, focusing on clean room environment, control air supply, that is HEPA filter, aseptic processing for non-terminal sterilized product, strict pressure differential in airlocks, unidirectional workflow to prevent contamination, and stringent quality control and sterility testing. Non-sterile products have their own requirement in separate part of Schedule M, but unlike sterile products, do not require the same level of control, airflow and aseptic condition within clean areas. Key requirement for sterile product, that is Part 2. Revised Schedule M required pharmaceutical manufacturer to adopt quality risk management, that is QRM, data integrity, enhanced facility validation and supply chain control to align with international standards like WHO TRS 1044 Annex 2. Specific requirements for sterile products include control clean room with airlock, proper air handling units for temperature and humidity control, and detailed guidelines for monitoring and maintaining these conditions. The revised schedule also mandates stricter measures for microbial contamination limit, equipment qualification, process validations, and establishes deadline for compliance, with a recent extension to December 31, 2025 for micro and small enterprises. Key requirement for sterile product under revised Schedule M: clean room standard, implement clean room with control airlock and maintain cleanness standard and filtered air supply, environmental control, ensure proper air handling unit, that is AHUs, and facility to control temperature RH, that is humidity, and air quality suitable for the product and operations. Microbial contamination, adhere to recommended limit for microbial contamination, airborne particles, comply with maximum permitted airborne particle concentration. Cleanliness, maintain high level of cleanliness within production and dispensing area. Clean room environment, manufacturing sterile product require dedicated clean room areas with a defined standard of cleanness, control air supply, that is HEPA filter, and appropriate pressure differential to prevent contamination. Aseptic conditions, operation involving sterile component or non-terminal sterilized product must be conducted under aseptic condition, often a grade A environment within clean areas. Airlock and pressure differential, entry and exit for personal equipment and material must be through airlock with interlocking doors, and pressure differential maintained to ensure a unidirectional flow from cleaner to less clean zone. preventing cross contamination. Separate area are needed for components of preparation, container closure, product preparation, filling, and sterilization to avoid any potential for cross contamination between operations. Personnel and equipment. Specialized personnel training for sterile manufacturing along with dedicated equipment is essential. Quality control. Robust quality control is critical, with sterility testing being a key aspect to ensure the final product is free from microbial contamination. Sterilization method. Manufacturing operations are categorized as either terminally sterilized or aseptic conducted, require specific controls for each processes. A comprehensive overview of requirement for pharmaceutical manufacturer to comply with the good manufacturing practices for sterilized product outlined in revised Schedule M. General consideration: clean areas. Sterile product must be manufactured in clean area with access control airlock, maintain appropriate cleanliness standard and filter air supply. Area separation. Separate area within the clean area for different operation, component preparation, product preparation, filling and sterilization. Manufacturing categories: terminally sterilized, product sterilized after filling, example autoclave. Aseptically processed without terminal sterilization, that is example sterile filtration. Quality control. Sterility testing. Sterility testing is the final control measure, not the sole assurance of sterility. Validate the sterility test for each product. Sample strategically, considering potential contamination risks, example beginning, end of aseptic fills, coolest part of heat sterilized load. Sterility assurance. Terminally sterilized, validate the sterilization cycle. Aseptically processed, validate through media fills. Simulated production with growth media, review batch record and environmental quality records. Endotoxin testing, monitor water for injection, intermediates and finished product for endotoxin. Rapid microbiological method, consider validated rapid methods for quicker results. Water environment bioburden, but with comparative assessment to pharmacopoeial method. Sanitization. Clean area sanitation, frequent and through cleaning with an approved written program. Use multiple type of disinfectants. Monitor for contamination and disinfectant effectiveness. Validate cleaning procedure to ensure disinfectant removal, disinfectant, detergent and handling. Monitor for microbial contamination. Store in previously cleaned container for defined period. Sterilize disinfectant, detergent. Use in grade A and grade B area before use. Sporicidal agent. Include sporicidal agent in the disinfectant program. Fumigation. Consider fumigation for reducing microbial contamination in hard to reach area. Manufacture of sterile preparation. Clean area classification. Classify clean area based on the required environmental characteristics, that is grade A to D. Use ISO standard for classification airborne particle concentration. HEPA filters. Install HEPA filters and conduct leakage tests average 6 to 12 months. Clean room classification. Classify clean room and clean air devices according to ISO standard. Differentiate between at rest and in operation states. Define maximum permitted airborne particle concentration for each grade. Particle monitoring. Routine monitoring of clean area while in operation. Grade A, frequent monitoring during critical processing. Grade B, monitoring frequency based on segregation from grade A. Use appropriate monitoring system, independent counter, network, etc. Demonstrate cleanup or recovery time after operation. Monitor for temperature and relative humidity. Microbiological monitoring. Monitor clean area for microbial contamination. Settle plate, air sampling, surface monitoring. Establish alert and action limit for microbial contamination. Grade selection. Select appropriate area grade based on nature of the process, operation and validation run, that is media fill. Terminally sterilized product, generally grade D preparation. Grade C filling. Aseptically prepared product, grade A with grade B background for critical operation. Processing. Minimize contamination. Take precaution to minimize contamination during all processing stages. Multi-product facility. Justify use of multi-product facility for live microorganism with effective containment and decontamination. Media fills. Validate aseptic processing through media fills. Simulate routine aseptic manufacturing step. Conduct 3 consecutive successful media fill initially and then repeat at defined interval, and after significant modification determine the number of container for media fill based on batch size, water quality, monitor water source, treatment equipment and treated water for chemical, biological contamination and endotoxin. Personal control. Minimize personal activity in clean area, especially during aseptic operations. Control personal movement. Component handling. Handle component bulk product containers and equipment carefully to avoid recontamination. Time limits. Minimize time between washing, drying and sterilization. Sterilization and use. Minimize time between solution preparation and sterilization. Filtration. Bioburden monitoring. Monitor bioburden before sterilization. Set working limit for bioburden before sterilization. Sterile equipment handling. Sterilize component, container and equipment. Pass sterilized item into clean area through double-ended sterilizers or other validated methods. Process validation. Validate the efficacy of new processing procedure. Sterilization. Terminal sterilization. Whenever possible, use terminal sterilization by heat in the final container. Sterilization method. Acceptable method: moist heat, dry heat, ionizing, radiation, ethylene oxide, filtration with aseptic filling. Sterilization. Validation. Validate all sterilization process, especially for complex formulation. Biological indicator. Use biological indicator as additional monitoring method. Sterilization record. Maintain detailed sterilization record for each run. Terminal sterilization record temperature and time accurately. Allow sufficient time for entire load to reach the required temperature. Take precaution against contamination during cooling. Monitor temperature and pressure independently. Validate automated control and monitoring system. Key requirement for non-sterile product. That other part. Journal GMP. Non-sterilized products are manufactured under Journal Good Manufacturing Practices outlined in other part of revised Schedule M, such as Part 1B for oral solids or Part 1C for oral liquids. Control environment. While still require a high standard of cleanliness, the environmental condition control are less stringent than for sterilized product. Do not necessitate the same level of HEPA filtration and air pressure differential. Prevention contamination. The primary focus is on preventing general contamination that could compromise the quality and safety of non-sterilized product. The primary distinction lies in the stringent requirement for maintaining aseptic condition, highly controlled air quality, that is HEPA filters, and strict pressure gradient for sterile product, which are necessary to prevent microbial growth and ensure product sterility. For non-sterile product, these advanced control are not required, though general GMP principles for cleanliness and contamination control remain vital. Thank you, thanks a lot, and keep watching our channel Quality Guru. Like, comment and subscribe please, more and more. Thank you.